and welcome to a new video on specific defenses, or as we might also call this, acquired or active immunity. And so what we're going to be looking at is the specific defenses, how they're able to defend against a very particular pathogen, how they're able to use lymphocytes, both T cells and B cells, to build or produce antibodies that defend against very specific diseases. So let's get started. So what we want to see is there are two main cells that are involved here, and these cells would be the T cells and the cells called the B cells. Now, T cells and B cells both respond very differently. First of all, T cells are cell-mediated immunity. And what do we mean by cell-mediated? Well, what I mean is they attack a cell that's either an abnormal cell or they attack pathogens that are inside of cells. They defend against these attacks. They're not really what we'd say producing antibodies, but they're usually going against either like abnormal cells, like cancer cells, virally infected cells, etc., things like that, or transplant tissues. Now, B cells, on the other hand, what these guys are antibody-mediated immunity. So, antibody-mediated immunity, and what I mean by antibody-mediated immunity is that these guys produce antibodies. We also said that this was humoral immunity because it goes into body fluids where we produce our antibodies. So there are a few things, and we mentioned in class, we mentioned in lectures, that there are innate immunities that you're born with. You can have genetics factors that you are just born immune with, already producing some of these antibodies, or you can acquire them either actively through getting sick or being given a vaccine, or very passively by getting it from your mother, either through the placenta, through mother's milk, or you're given an antibody sometime while you're ill. And all of these would help me do that. So what we're going to now see is with these T cells, how they interplay with some things called a major histocompatibility complex, right? So we have our MHC molecules. MHCs, the major histocompatibility complexes. And these are glycoproteins, and they're on the cell surface of many cells. And they come in two types, right? They are either MHC class 1 proteins or MHC class 2. And MHC1s are found on all nucleated cells, right? Whereas MHC2s are only found on antigen-presenting cells, abbreviated APCs. And they also have, they pair to a particular type of T cell, T cells with a very specific marker on it. The MHC1 proteins are only able to interact with CD8 T cells. CD8 T cells. Let me erase that and start that over. CD8 T cells. Whereas the MHC2s do CD4 T cells. And these are the cells in which they have the CD4 markers on it. And just remember the equals 8 rule. When I say equals 8 rule, let's think of this. MHC1, 1 times blank equals 8. Well, 1 times 8 equals 8, CD8. 2 times blank equals 8, right? Because MHC2, right? 2 times blank equals 8. Well, CD4 T cells, right? That is very important that you do keep up with which MHC goes to which particular type of T cell. So the first thing we're going to really look at is take a look at MHC1s and how they play a role in your particular immunity. So let's just take a look in our body, and we have a cell here, and this cell in my body is like any other cell. It has a nucleus, and in the nucleus there are DNA, and the DNA codes for proteins in my body. But in this case, let's say I've got a thing that comes in here, a virus, and this virus, like all viruses, contains some nucleic acid in it, right? DNA or RNA, and it will insert that DNA or RNA into the body, right? This nucleic acid. And this nucleic acid will enter into your nucleus, for example, and hijack your cell's transcription and translation or replication machinery, and it will end up, this virus now will start making this cell here start producing proteins that are now viral proteins. It will start producing viral proteins, in which we call these proteins endogenous proteins. These are the endogenous proteins. Now what happens is that your body will then begin to produce a molecule, and let's say we grab that guy here and display it out, called an MHC1. Because remember, all nucleated cells possess them. And it will take this antigen here and display it on the surface as endogenous protein or antigen. Antigens are anything that can cause immune response. And now that it's out there, think of this like a red flag, right? This displayed antigen here, right, will now be able to interact with T cells. And not just any T cell, but a CD8 T cell. And this CD8 T cell possesses with it, guys, a T cell marker, right, T cell receptor, and a CD8 protein marker, little CD8 protein there, marker that's on it, making it a CD8 T cell. And he is going to begin to inspect this antigen, and what it could do is this CD8 T cell could destroy, could release perforin, for example, destroying the cell, right? It could destroy the cell membrane by releasing perforin. It could release a chemical molecule that make this cell undergo apoptosis, or it could do some other things to destroy its metabolism, so to speak. But the big thing is here that we have these cells, the CD8 T cell, going to this cell who's infected with a virus, and what will happen is either this is a memory T cell, for example. Let's say he was a memory T cell, and he has seen this antigen right here before, then he will know what to do about it. Now, let's just say he's not, he's an inactive T cell, just an inactive T cell. He may go in and get this and then be able to do something about it at that exact moment, right? But if he's also going to help stimulate some long-term responses by remembering those cells. Remember, with T cells, there's helper T cells, memory T cells, cytotoxic T cells, and suppressor T cells. Help me catch some. Because this T cell is going to find this antigen here and say, help me catch some of that antigen and help me attack it if I ever see it. He's going to either attack the virus-infected cells or take this cell to a B cell. That's the two things they can do. So that's what CD8 T cells help me to do, is detect the cell who's infected with the virus and try to destroy it. Now, it's possible that not every cell out there is actively producing proteins who's infected with the cells. Say, for example, herpes, or some of these viruses that follow the lysogenic life cycle of viruses, but that is neither here nor there. We just will make sure we know that's how they behave when they are actively displayed, which is going to be the case at some point. So let's take a look now at another interesting thing, and on this one we need to have our macrophages. And so here I have a macrophage, which we're going to refer to as being an antigen presenting cell, right? Let's draw a nucleus so we know that this guy does in fact have a nucleus here. And now let's have us a pathogen. And my pathogen here, a bacterium, and let's draw some antigens on it, right? A circle antigen, a triangle antigen, let's do a square antigen, and let's do an X antigen, right? Let's do some different antigens. So these are the antigens. Now the goal here is I want to make an antibody against it, right, is what I want to do. My goal is to make an antibody against this antigen if we can. So let's use circle antigen. That's the one I want to make. That's the one that's going to be really good. So let's hope this works. So how does this happen? So an antigen presenting cell is going to be phagocytic. And it's going to phagocytize my little bacterium here. It gets phagocytized. It's brought in, right, with its antigen. So let's get the antigen, square antigen, right, circle antigen. Let's get X antigen and triangle antigen there, right? And so what happens is a cell, being a macrophage, produces lysosomes with lysosomal enzymes, right? And with lysosomes, what they are going to do is they will fuse with this phagosome, releasing its lysosomal enzymes, and it will begin to degrade up this cell, right? Leaving behind some antigens, right? Leaving behind parts of this cell here, leaving behind only the antigens. Now, what we're going to do is our nucleus will produce an MHC protein. It's going to come by and grab an MHC. Now, this is an MHC2 protein, and it is going to take this antigen, bring it out, and display it, right? It is now displayed. Or now with its being displayed, it's extracted, then it's bound to the cell membrane. Now here's where things get very important, is if I've got a T cell that comes in here, an unactivated T cell, who is a CD4 T cell, he's got a T cell receptor for the antigen, but he's got a protein called a CD4 marker on him, makes him a CD4 T cell. So his T cell receptor here, T cells can only recognize antigens when they are displayed on the antibody, on the MHC. So let us take a look at this and kind of use an analogy. Let's think of this like it was a Cinderella story, right? Cinderella is our bacterium, our disease. Doesn't that sound like, oh, I've got Cinderella, I've got the disease, right? Cinderella disease. Well, Cinderella comes in, she wants to dance with a Prince Charming, right? The antigen presenting cell. She comes in, but the clock strikes midnight, right? Her carriage turns back into a pumpkin, and she drops the glass slipper, the antigen, right? And Prince Charming has to pick up the antigen and go around and find a young lady in the land that fits it, right? So he takes the antigen. Now, CD4 T cells are like you young ladies out there. And I know that every one of you know what you exactly expect for when a man is going to get down on his knees. He's going to lift up that jewelry, that box, that ring box, and he went to Tiffany's and bought that diamond, and you're waiting on that. And you know precisely what it is how you want that. And he gets down on the knees, calls you by your full name, asks you to marry him, right? And only if he does it that way. Now, if he just goes in and says, well, maybe I'll spell out, will you marry me, in her alphabet cereal, you're like, no, that's not good enough. I don't want it. See, a CD4 T cell can only recognize this antigen when it is displayed on a major histocompatibility complex. It's got to be on the MHC protein. And it's an MHC2. 2 times 4 equals 8. Equals 8 rule. Now let's take this CD4 T cell and see what he's going to do. Now, This antigen can either be attached to a dendritic cell, right? And that can stimulate lymphocytes or some kind of macrophage. Any cell that is acting as what is referred to as an antigen presenting cell, right? Because it presents the antigen to the lymphocyte. So let's follow him, right? Now, what is going to happen here is when it goes to the T cell, this T cell is going to get activated to become an activated T cell, and it's going to basically become a T helper cell. So it goes become a T helper cell because we've given it the antigen, right? So let's take my CD4 cell here, right? CD4 T cell, who is a T helper cell, who has been sensitized to my antigen. He is active, right? He is now completely an active cell. Now, he could become a T helper cell, yes, right? He also become a memory T cell, which also, so he can member a memory T cell, right, or a helper T cell. And that's what he can become here when he's activated. Now, when it comes to the previous action, this cell here could actually become a cytotoxic T cell. This cell can become a cytotoxic, a memory, or a suppressor T cell after this action. So that's also important to keep straight. So let's take this CD4 T cell. Now, CD4 T cells, think of this, guys. Let's continue our Cinderella story. Now, you ladies, when you are being proposed to, the first person you're going to call is going to be Mama, right? You're going to call Mama. Well, Cinderella's mama was an evil stepmother. She was bad. She was a B cell. She's bad, right? The bad B cell. And what we're going to do is take these B cells, and on this B cell there is a very specific protein, a very specific type of antibody that's on its surface, right? And these antibodies here, this is an IgD, right? Now, what we know is that our cell here, our CD8 or CD4 T cell here, he's got my antigen, right? My circle antigen, right? He's got the CD8 marker here, right? And what he's doing is he will take this and it will give it and bind to the IgD, acts as a receptor for my antigen. Now, what happens is this, if there is an IgD who has a receptor for that antigen, the antigen shows up, either the B cell brings it over, or the T cell brings it over, or it just shows up in the body. It can bind to that, and it gets internalized. And what will then happen is this cell will then produce an MHC class II protein, that it will then display that. And once this happens, we say this B cell is sensitized. And when it's sensitized, right, it becomes either a memory B cell, or a plasma cell. And when it becomes a plasma cell, it will make the antibodies against only that antigen there. Now, it's sensitized, but it doesn't really activate again until a helper T cell comes in and stimulates its activation. It turns on and will continue on to make some cells. Now, here it's important to note that B cells can recognize these antigens in their native form. They do not have to be presented in that respect, right? So, as we said, you might have my T cell bringing the antigen in. You might have the antigen showing up. The antigen shows up, right, with a T cell bringing in with the T cell receptor and that. They will activate it when they bind to this, when the helper T cell binds to this, right? as you'll see, this guy, somehow, this T cell who's extracted this antigen, it's got to get there to the B cell somehow. It can get there in the body by just getting sick, etc., 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 right? As long as it gets there. But it will get there, it will sensitize it, and that's when my helper T cell will bind to this MHC, activate this in the memory B cells or the plasma cells. And this is how we end up making, finally, from the plasma cells, an IgA that only responds to that antigen. That's all it can respond to, would be circle antigen. It wouldn't be able to respond to any others. So either it's on the surface of a bacterium or it's floating around out in solutions. This antibody will attack it. And so that is how we ultimately make antibodies that are very specific and how specific defenses work. I really hope you found this video helpful. Please comment, like, and subscribe to see more videos like this. If there's anything else I can do to help you, if you have any questions, please don't hesitate to email me. Send me some suggestions on what you'd like to see in the future. Thank you so much for all your support, and I'll see you guys in the next video. Thank you so much.